Hi ladies, this is chapter 14 on protein synthesis. So the first thing I'm going to talk about is the flow of genetic information. So basically with gene expression, this is the process by which DNA directs protein synthesis. So this is going to have two stages, transcription and translation. Transcription is turning DNA into RNA, and translation is using RNA to make a protein. So here's a little bit more into transcription and translation. So transcription is the synthesis of messenger RNA using information in DNA. So DNA is going to be the template. So if you open up this DNA strand, you can use that as a template for transcription, and the mRNA will be a complement of that DNA sequence. With translation, this is going to be the synthesis of a polypeptide by ribosomes, and this is going to be using the information from the mRNA strand, so making the amino acid sequence that corresponds to the mRNA sequence. Before we get started, I'm just going to do a quick comparison of RNA and DNA. So RNA has nucleotides just like DNA. The only difference, there's three differences. The first one is that ribose sugar is used instead of deoxyribose sugar in DNA. RNA is typically single-stranded. DNA is typically double-stranded. And you'll have one nitrogenous base is different in RNA. So uracil will replace thymine. Okay, uracil will can bond to adenine during transcription in order to copy the code. Eukaryotic and prokaryotic cells go about the process a little bit different. So in eukaryotic cells, this process is separate. So transcription is going to happen in the nucleus, inside the nuclear envelope, and translation is going to happen out in the cytoplasm. With prokaryotic cells, what's really interesting is that transcription and translation are pretty much going to be happening simultaneously. As the transcript is made, the translation, the ribosomes will be copying that and making that protein right next to the DNA because there's no segregation. In eukaryotic cells, there's also a little bit of modification of the mRNA. The first thing I'm going to go into in detail is transcription. So here's a little bit of vocab that I'm going to be referencing as we go through. So the first thing I want to show you is how transcription begins. So transcription requires a promoter. So this is an example in a eukaryotic cell, but there's so within eukaryotic cells, there's these things called Tata boxes that are going to be at the very beginning of the promoter. And the promoter is just a sequence of DNA that basically tells the RNA polymerase where to start making mRNA. Transcription factors are very important in this part, and we're going to look at regulation a little bit later, but transcription factors will bind to the DNA, and basically they help the RNA polymerase copy the DNA and make an mRNA transcript. So here are the three steps of transcription. So there's initiation, elongation, and termination. Initiation is the binding of the RNA polymerase with the help of transcription factors at the promoter region. Elongation is when the RNA transcript is going to begin to start. So it always goes in a 5' to 3' direction. That's how RNA polymerase can add these mRNA nucleotides. And then eventually a complete RNA transcript will be created and the sequence will be terminated. You can see here the direction of transcription. The new RNA is made in the 5' to 3' direction. And the way that this works is that it uses the DNA basically as a template. The template strand of DNA is right here, and you can see that there are complementary base pairs. C with G, U with A with T here, complementary base pairs in order to make an exact replica of the gene of interest on the DNA. And note that this RNA polymerase is copying one side of the DNA molecule. There could be a completely different gene on the opposite side of the DNA. Once the mRNA is created, there can be some processing. This is called RNA processing. A couple of things that could happen are a GTP cap is added at the 5' end of the mRNA sequence. Sometimes there's a poly A tail, so a lot of little adenines added. This is at the 3' end of the mRNA. Remember, these primes just are associated with the carbon in the nucleotide, so the 5' part of the ribose sugar would be closer to the end, and that's how it's labeled. Another thing that can happen is RNA splicing, so removing introns and joining exons together. Introns are basically non-coding regions of the gene and eventually the mRNA. And so they can be removed, and then the exons are the coding regions. What's really interesting is that the exons can be arranged in a variety of different patterns. So you can see here, this is called alternative splicing. Splicing together exon 1, 2, and 4 can produce protein A, or splicing together 1, 3, and 5 can make protein B. So you can get a lot of different results. And spliceozymes are the special little enzymes that help facilitate this. So what's really interesting is that there is a universal genetic code. So this is how all organisms can share the same 20 amino acids and 4 nucleotide bases. So if you have a gene in one organism, you can put that gene into another organism and they have the machinery to express it. So you can see here tobacco plants expressing a firefly gene, a pig's expressing a jellyfish gene. We can express pretty much any gene because we have we have a way to produce protein. So if we have the DNA, we have the machinery in order to take that DNA and produce that protein. So this is the universal genetic code, and it's based on mRNA triplets. We call these codons, and they correspond to a specific amino acid. So when you're trying to figure out what the final protein is going to be from a transcribed mRNA sequence, what you have to do is use this genetic code table. So the way that you use it is the first mRNA base, you will, in the triplet, you go here. Say, for example, the first mRNA base is A. So you go here. So the second mRNA base, maybe it's U. So you'd go to this column. Third mRNA base is G. So you go to this one, and this actually is AUG, which is the start codon. Very important one that I will talk about very shortly. But that's how you can use this little codon table to determine what mRNA sequence, which codon corresponds to which amino acid. And you can see here, here are the amino acids and what each of the codons codes for. So for example, all of these little guys code for leucine. All of these ones right here code for proline. And you can see there is a little bit of replication. So there are multiple codons that code for the same amino acid. The last part of RNA synthesis is translation. So here's a little bit of vocab with the two different types of RNA that we're going to be focusing on. We already looked at mRNA. The next two are transfer RNA and ribosomal RNA. Transfer RNA basically transfers amino acids to the growing peptide in a ribosome, and it does this by using an anticodon. So this anticodon will base pair with the mRNA codon. I'll show you that in a second. Ribosomal RNA facilitates specific coupling of the tRNA anticodons with the mRNA codons during protein synthesis. So it's kind of helps organize everything. And it is made up of large and small ribosomal subunits. So here is tRNA and ribosomal RNA, or what we usually refer to as a ribosome. You can see here up here with the tRNA, the reason it has an anticodon right here. And this anticodon is complementary to the codon in the mRNA. At the very top, there is an amino acid attachment site. And this is where the amino acid will be attached until it is removed from the tRNA molecule. Down here is the ribosome. You can see that the ribosome is made up of a couple of different sites. You have the E site, the P site, and the A site. The A site is where new tRNA will carry in the new amino acid. The P site is kind of where the reaction is going to happen to catalyze the attachment of the new amino acid. And the E site is the exit site. Okay, the mRNA is going to go right in between the small and the large subunit. So when we're building a polypeptide, there are 3 stages of translation: initiation, elongation, and termination. And all 3 of these stages require protein factors to help aid, and also energy. Here's initiation. You can see here that this starts at the start codon. So AUG is present on the mRNA. You can see you can see the anticodon on top is opposite of that. The first initiator tRNA always has methionine, so that's how all proteins start. And once this mRNA is bound to the small ribosomal unit and the initiator tRNA is attached, the large ribosomal subunit will come in. This will happen at the P site. From here, elongation will take place. So the first tRNA will come in that corresponds with the anticodon corresponding to the codon, and that'll come in at the A site. A reaction with GTP will facilitate the addition of this amino acid to the sequence. From here, you can see the peptide bond formed. From here, there'll be a little shift, and the mRNA will shift, and everything will move into the next site. So what was in the A will now be in the P, and whatever enters the E, that's the exit site. So that tRNA can now leave, and it's not attached to the protein anymore because of the peptide bond that was created here. This makes room for a new tRNA molecule to come into the A site. And when it comes into the A site, you'll repeat the same process all over. Okay? Adding a new peptide bond to connect these 2 amino acids together. There'll be a shift, and the old tRNA that doesn't have the amino acid attached to it anymore will leave, and the process will repeat. Finally, termination will take place when a stop codon occurs. So because the stop codon codes for a release factor, and this release factor will allow the polypeptide to be free, and it will allow the mRNA to be released and the subunits to separate from each other. This isn't the end, so the protein will also have to be folded, and it might have some post-translational modifications. Post-translational modifications include possibly attaching a signal peptide and using an SRP to bind to that signal molecule, that signal peptide, in order to make sure the protein makes its way to the correct location. So you can see here folding. I'm going to talk about this one first. You can see here the SRP allows the protein to enter into the correct place. So in this example, it's going to enter into the endoplasmic reticulum, so it goes to the right place, and usually these molecules that are here will then be secreted outside of the cell and packaged appropriately. When proteins are made, they need to be folded correctly as well. So this is showing the primary, secondary, tertiary, and quaternary structure from the beginning of the year. Make sure you review these. Remember that secondary structure has to do with the beta pleated sheets and the alpha helices, and tertiaries is just the folded polypeptide with those little R groups and how they interact, and quaternary structure has to do when there's 2 or more polypeptide chains. To review, make sure that you watch the video on protein synthesis and bring any questions you have with you to class. Have a nice day, girls.